Good morning, everyone. It's so nice to see so many people here at 8 in the morning after a late night with the Sage Against the Machine, or whatever you were up to last night. Welcome to the second half of the session, Innovation in California Herbaria and Specimen Records Use. Today's session is a celebration of the transition of your herbaria into the digital age. And in fact, there are two or three more talks tomorrow afternoon in the Fur Room in the Floristics 2 session if you can't get enough this morning. So I'm the first speaker. I'm Alison Colwell. I'm the curator of the UC Davis Center for Plant Diversity Herbarium. And I've been there 2 years, and most of you know Ellen Dean, who's here this morning, who is my predecessor. So I'm following in her footsteps. My herbarium is about 320,000 specimens. Most of our California specimens are digitized, and we have about 88,000 of them imaged. Okay, so first slide is, here we go, harnessing the power of inadvertent collecting. And my co-authors are all entomologists, and Emily Meineke is a junior faculty, just started 2 years ago at the beginning of the pandemic with me, and I guess this talk could be titled What I Did with My Pandemic Summer. She has a new grad student, Grace Horn, is in the thick of analyzing the data we gathered in our pandemic summer, and Dave Eng was a junior specialist it's a program for recent undergraduates who get training in science before they land a job. So Emily came to the herbarium looking for specimens she'd heard about that were collected by an entomologist, Ian Pierce, 13 years ago, that were all about insect herbivory. They were insect herbivory vouchers. And she, in the process, we cooked up a study on bias in botanical collecting to support her research. Emily uses herbarium specimens to answer large questions in insect ecology and evolution. Her work is a good example of the novel uses that large online collections of specimens are being put to. Insect herbivores have important effects on plant fitness and productivity. The so-called arms race between the two groups since the dawn of land plants is thought to have spurred the diversification of both groups. Insect damage to leaves is one of the proxies that entomologists use to measure the effects of insect herbivores on plants. And many inferences can be drawn from the patterns and the extent of the damage. For instance, Emily is studying how light and vehicle pollution affects insect behavior as measured in herbivory on street trees. So each of these photos shows a different type of herbivory on fossilized leaves from the Paleocene-Eocene thermal maximum, a time period when air temperature warmed 5 degrees Celsius in the span of 10,000 years. This time period is arguably one of the closest analogs in the deep past to recent climate change. Such fossils show us that during this period of rapid warming, herbivory became more abundant and diverse. You can see many different types of herbivory damage present on these leaves. And it was largely a result of insect range shifts. In contrast to such predictions from the fossil record, modern studies suggest that insect biomass might be in severe decline. The insect apocalypse, as we call it, is making it very difficult to understand how insects are responding to global change. Unfortunately, data on insect herbivores are severely limited over time, over geography, and over phylogeny. For instance, this graph shows a histogram of herbivory study duration, with study count on the vertical axis and study duration on the horizontal axis. The vast majority of herbivory studies are between 1 and 2 years long, which means they don't begin to cover the period of anthropogenic climate warming, and most were conducted since the 1950s. Insect herbivory is important to agriculture, to global productivity, and feedbacks to the atmosphere, so there's a great need to find sources of historical data, and that's where herbarium specimens entered into Emily's work. Herbarium specimens, such as these 4, are potentially the key to filling this data gap, as they have been collected around the world for now about 400 years. These 4 specimens show 4 types of insect herbivory: chewing, stippling, little points from sucking insects, leaf mining, and leaf skeletonization. So each is characteristic of different insect groups, which are often identifiable to genus from the damage they cause. While these herbarium specimens weren't collected expressly for studying herbivory, they are nonetheless providing crucial data about insects. Here's an example of the use of herbarium specimens for insect ecological data. Examination of a time series of herbarium specimens from 1896 to 2008 for herbivory. On this graph, the x-axis is the year the specimen was made. On the y-axis is the probability that any specimen showed signs of damage. So each dot represents one of 4 species, and the dots at the top where damage was present, at the bottom where damage was absent, and the graphs show that overall the presence increased, implying that 23% increase of herbivory occurred between 1896 and 2008, with the gray being the confidence intervals. But here's the rub. Because this is a use that collectors never envisioned and consequently did not address explicitly or even consciously, Emily's work has been criticized that implicit or unconscious bias may affect conclusions drawn from the work. Why is this not advancing? All right. So, biases. We collectors have all kinds of biases that we know about. Undersampling. We like to collect plants that fit on an herbarium sheet. We have favorite taxa, and we all go out in the spring. Yes, we know about this. And some of these can be controlled for by the label data. But bias, such as insect damage on the plant sample, is a biggie that is hard to get at. In addition, there's preservation bias in herbaria as an added layer. Often the curator and mounters make decisions about what is mounted and or retained or discarded. So the bias at that level of specimen processing needs to be looked at also. So we set out to see if we could quantitate or quantify collector bias. We asked, does the collector bias for herbivory vary across taxa? Does it swamp out existing differences in actual herbivory across plant taxa? In other words, what is the direction of the bias, the extent of the bias, the consistency of the bias? And in this box shows herbivory on the x-axis estimated randomly on the actual plant, and the y-axis estimated from the herbarium sample. And if collectors were not biased, there would be a one-to-one line of the specimen matching the actual plant. What we expect is that there's a down bias on herbarium specimens, and just between you and me, we collectors are a little biased against blemishes, including insect damage. So someone comparing the source plants in our specimens, we would expect them to see less herbivory on our specimens, resulting in this lower slope. But we expect the down bias would probably be more or less the same across all of our specimens. So we might avoid insect damage to some extent. So Emily and I took advantage of an empty campus and some quiet time to set up a study to examine these biases. This is the UC Davis campus, and you can see Interstate 80 along the south side and 113 on the west, and through campus flows Poudre Creek, and that's encompassed by the UC Davis Arboretum, a living collection and demonstration landscape of climate-durable plantings, and many of these are native species, and they're planted along pathways on either side of the creek, and there's parking lots and paths and restrooms. In other words, a great site for an outdoor pandemic group activity. So we chose native species because they have diverse insect predators. We chose shrubs and trees that were numerous enough to be and accessible enough for volunteers to collect in a morning. We're dealing with 100-degree temperatures in the middle of the day. We chose chewing and mining because they're easy to see. And we did Dave Eng did this, pre-surveyed all of the study plants for herbivore damage, and Ian Pierce is here showing you how you can do this on shrubs and trees with shooting the tree with your finger, and whatever leaf your finger lands on, you assess that leaf, and you repeat this many times on each cardinal direction of the tree, and you get an overall estimate of the herbivory, and sometimes this involves ladders. The finger method covers presence and absence of herbivory and percent damage to give an overall estimate. Then we recruited volunteers from all experience levels to make their specimens from the pre-sampled trees and shrubs. This was a great year to do it. There was 30,000 fewer students learning to ride bikes on these paths because the campus was closed, and lots of volunteers at both the Arboretum and the Herbarium eager to do the work. This was a blind study in that everyone received the same, as you see, like socially distanced instruction, and they did not know why the study was being done. Emily was a stealth professor. She had just arrived at the campus. They didn't know what she was up to. So everyone who volunteered filled out an exit survey, self-evaluating their expertise in plant collecting and self-evaluating the important features, including insect damage that they might have used in their selection process. Here's a very pleased Dave Eng displaying one day's volunteer collection effort. We held 5 collecting days, ended up with almost 800 specimens. And on the right, a rather less pleased looking Dave grinding through the quantification of the herbivory. It took them actually this entire past year to do. And you can see some of the specimens, they're all over the lab. They now have an herbarium cabinet. So results, here are our results. This is chewing results, and on the left is the June results, and on the right is the August results. Those of you who've had 2 cups of coffee will notice that there's not 14 specimens because this was a hot year and there was a lot of, the ribes viburnifolium dropped all its leaves before August, so we had to drop that. What you can see is that the left-hand bars are all the ecological samples, and on the right are the herbarium samples. And you can see that there's much more herbivory overall in August, and you can see consistently that every single pair, that even in both June and August, there's less herbivory on the specimens. So very consistent down bias across species. Nice to know this or verify this. Leaf mining, only a few groups, Symphoriocarpus, Quercus, and Populus showed leaf mining, but again, those that did, the herbarium specimens consistently showed much less damage. Another way of looking at this is the box graph that I had showed you before. So percent leaf area removed on the plant is across the bottom. Percent leaf area seen removed on the herbarium sample is on the y-axis. And you have June graph and August graph. And you can see on the left that collectors were less selective in June when there was less damage. That's essentially a one-to-one line among all the species, each point representing a species, and then the little X's around them is the variance and standard deviation. But they were more selective in August when the damage was heavier. And for mining, collectors were less selective overall, probably because it's not as obvious damage. And the really big damaged plant, the Porcymphoriocarpus, people definitely were very choosy about those heavily damaged specimens. So again, the pattern holds more or less for all of these species. Curators and mounters. We presented the specimens to our curators and mounters, and they would mark whether they would use the specimen, and if not, why not. And we have not noticed much bias at that level. It's not analyzed yet, but maybe there's less choice. Maybe it's not as obvious. So the question posed to us from this Stigmella moth larvae is, what do we botanists do about our biases against insects? And when I asked Emily if I should propose to this audience that botanists change their collecting methods, she was emphatically against it, because any change in our bias now would complicate her work, muddying the picture she has worked so hard to get about our botanist bias. Instead, she suggests that if we do make a specimen documenting interesting insect use, that this should be stated on the label. And stating the purpose of a specimen functions both as an alert to the world of the existence of an insect documentation, because we put all of our specimen data into Symbiota where people can search on such things, and it allows for its exclusion in a study like this. So acknowledgements, a huge number of people to get this kind of data. There's 40 volunteers and the various labs involved with the analysis. So thank you. And we have 1 minute for questions. Yes, Mike.
How often can you actually determine what the insect species is, or is it generally just the group of insects? That would be valuable data.
So unless the insect died inside its chamber, usually it's to genus, and even more often to family. And entomologists are really good at this. And I'm not, but yeah, genus is like, for instance, the genus of moth, they make a particular question mark pattern. So.
Is that data that should be reported?
Oh, yeah, I suppose. All right, so the question was whether this data about what species of insect is, should that be recorded? Now, I suppose, yeah, that if you can. I mean, if I see a gall or some other thing and I record or make a specimen out of it, it would be nice if I went over and asked an entomologist about it. Of course, that's a bit more work, and is that realistic? But at the very least, I could say, this is a gall, or this is some leaf mining, or something like, or say something about the prevalence. Yeah, and if I put that on my already somewhat verbose specimen labels, somebody might appreciate that in the future. Yes? Well, we actually put these through a dryer. It was very hot, so probably that would have killed a lot of things, and then they went straight into their lab. Ordinarily, we would freeze them right away afterwards, but this is, way too much for our freezers, so it went back to the entomology lab, and they started analyzing them right away. So I suppose there could have been more, but the larvae, having been in paper in a hot dryer on my back porch in 100+ degree heat, I don't think there would have been much, but yeah, we did not actually control for that. And I should probably introduce our next speaker, Natalie Love. Natalie, are you here? Ah, there you are. All right, welcome, Natalie. Here's your first, I'll just say that you are a Frost postdoctoral fellow at Cal Poly, San Luis Obispo, where Natalie's research focuses on urban forestry in California. She received her PhD from UC Santa Barbara, where she studied the influence of climate on reproductive trait evolution using California jewel flowers striptanthus, and I guess we're gonna hear a bit about that. Natalie uses the Robert F. Hoover Herbarium, and that has 100,000 specimens, and they are all imaged.
Thank you. Hi, everyone. Good morning. My name is Natalie again, and I'm a Frost postdoctoral fellow at Cal Poly. I work now mostly on urban forestry, but I'm going to spend a little bit of time today talking about some of the work that I did for my PhD with Dr. Susan Mazur at UCSB. And we studied how climate and climate change affects the reproductive phenology of the jewel flowers. And you could see that title if it showed up. Hopefully that is not a indication of the rest of my presentation today. But if it is, we'll make it through it, and I'll walk you through anything that is a little bit messed up. All right, so a major question in ecology is how will climate change affect plant phenology? Because changes in plant phenology, whether that's the timing of leaf out, the timing of senescence, the timing of flowering, the timing of fruiting, changes in those types of events can have major ecosystem-wide consequences. So changing in the timing of flowering, for example, can affect the relationship between plants and their pollinators, especially if plants and their pollinators are not responding to changes in climate, like increases in temperature, in the same direction. It can also affect plant-plant interactions, such as competition for soil or for pollinator resources. And it could also potentially expose plants to novel environments, such as drought or frost, or even novel herbivores. And therefore, predicting how plant phenology might change in the future or assessing how it's already changed really is the first step in trying to understand some of these broader ecosystem-wide consequences of changes in phenology on ecosystems. And one way that we can predict how species will respond in the future is to start by assessing and evaluating how species respond to current variation in climate across their range. And so what we can do is look at the relationship between some kind of onset of a phenological event, which is shown there on the y-axis. In this case, I'm showing flowering date, and how that relates to variation in some climatic variable across the range of that species. In this case, we're looking at spring temperature. And each one of those points on that plot represents some kind of phenological observation. And there are different ways that we can get phenological observations. We can get phenological observations from large phenological networks like the National Phenology Network or NEON. We can get phenological observations from field studies or from common garden experiments, or we can get phenological observations from herbarium specimens. And we can use those observations to actually quantify the relationship in a phenoclimatic model, the relationship between the onset of some phenological event and some climate variable. And the slope of that relationship gives us how sensitive a given species is to variation in some climatic variable. So in this case, the slope of this relationship tells us that for every 1 degree increase in temperature, flowering time for the species advances about 4 days or so. So we can use these types of relationships to assess how sensitive species are to changes in temperature or any climate variable of interest. And if we're talking about using herbarium specimens provide at least 100 years of record in California, and especially for my species, and we can look at how phenological events have actually changed through time. So in this case, each one of those dots is an herbarium specimen collected across the past 100 years, and we can look at how phenological events have already been impacted by climate change using these types of models. And the slope of this relationship in this case tells us that for in the past 100 years, plants have been collected on average about 10 days earlier. So these are some ways that we can use phenological observations across space and across time to assess how plants are going to respond to climate change in the future. Oops. So my talk today focuses on investigating phenological relationships between flowering date and climate using the California jewel flower as a study system. And the California jewel flower, Streptanthus tortuosus, is really ideal for studying these types of questions for a few reasons. One, the species itself is very widely distributed and broadly distributed in California. It occurs in a very wide range of habitats: coastal populations, low elevation, hot, dry foothill populations, all the way up into the alpine. So it ranges from about 60 feet or so all the way up to almost 10,000 feet in elevation. So it spans a really broad climatic gradient, and therefore we might be able to expect to detect relationships between flowering date and climate. And second, Streptanthus tortuosus is very well represented by herbarium specimens. There are over 1,000 herbarium specimens that are now imaged, and they span over about 100 years of a collection period. And so that gives us a very, very powerful system to assess phenology, not only through space, but also through time. So it's a very, very powerful system for trying to understand the relationship between phenology and climate change. I was specifically interested in this project in three main questions. First, has flowering phenology changed in the past 100 years? So have we captured a shift in phenology in the past 100 years in herbarium specimens? Is there some kind of evidence of phenological shift preserved in herbarium specimens? And second, because the species has a very, very broad range, I was interested in trying to understand whether those shifts were consistent across the entire range of the species. Are some populations shifting faster than other populations in magnitude or in direction? And I was specifically interested in comparing warm versus cool regions of the species range. So populations that occur at low elevation warm areas versus populations that occur at higher elevation cool areas. And if we are able to detect these types of divergent shifts, there's kind of two potential explanations. Either populations in one area are more sensitive to temperature, or populations in one area have experienced a different degree of warming. So we were really interested in testing whether or not shifts were related to phenological sensitivity to climate variables, specifically temperature, or changes in temperature. So herbarium specimens provide this great snapshot of phenology through space and time. And so I wanted to start by talking a little bit about how we can actually use herbarium specimens to study phenology. So first, all of the information that we need is right on the label. This is one of the specimens that I used in my data set. This is one of Alice Eastwood's collections of Streptanthus tortuosus from Yosemite. And on that label, of course, there is a date of collection. There is both a day of year of collection, or the DOI, which ranges from 1 to 365. And then that is oftentimes used as a proxy for flowering date in these types of study, the day of year of collection. And that is what I use for my study as well. There's also a year of collection there as well, right? So we know exactly what year this plant was collected. This was collected in 1923. And then there's a location description as well. And so we can use that location description and georeference it, turn it into a set of coordinates, and then use that information to characterize the climate that that plant was experiencing at the time it was growing in Yosemite. All right, this one got a little bit messed up. So in my study specifically, we used about 750 imaged herbarium specimens, and those herbarium specimens were collected between 1902 and 2013 across a really broad climatic gradient in California. And what we did was we extracted the day of year of collection from each one of those specimens. We georeferenced them, so we assigned coordinates to each one of them, and then we used that information to extract a wide range of climatic variables in order to understand how phenology responds to those variables. Today, I'm specifically going to be focusing on spring and summer temperatures. That is the season in which this species is actively flowering, and it is representative of the general patterns that we found in response to phenology. So because we were interested in understanding whether or not there were phenological shifts and whether or not those shifts were consistent, we looked at the effect of year on the day of year of collection, and we used a linear regression to do this, and we included this year by temperature region interaction to ask whether the effective year was different between the temperature regions. And the way that we defined these temperature regions, you can see up on the map that some of those observations are colored red and some of the observations are colored blue. So we actually took our entire data set, we divided it into two based on the median mean annual temperature, and then we removed the middle 20% so we could get this very strong comparison between warm and cool regions and understand the relationship between phenology in those two regions of the species range. We also constructed a linear regression to look at the effect of climate on the day of year of collection. Specifically, we were interested in the effect of climate anomalies, so deviations from annual temperatures. How do plants respond to warmer or cooler than average years? And again, we included this climate by temperature region interaction to ask whether phenological sensitivity to temperature differed between two regions. Okay, so let's take a look at some of the results. So here we are looking at the relationship between the day of year of collection, which is on the y-axis there, and the year. So we're looking to see, is there evidence of phenological shifts in the day of year of collection or in flowering time through time? And what we found is across the entire range of the species, so this is looking at all of the specimens together, there's been about a 10-day advancement in the day of year of collection. So on average, specimens are getting collected about 10 days earlier now than they were 100 years ago. However, we were really interested to understand whether these shifts were consistent across the entire range of the species. So are populations or individuals in warm regions shifting their phenology in the same direction or even at the same rate as individuals in cool regions? And when we look at that, what we see is there is a difference. We did, in fact, detect divergent shifts in phenology where individuals in warm regions are advancing their flowering date about 20 days relative to 100 years ago, and we didn't actually find any evidence of a significant shift in flowering date for individuals in cool regions. So what this means is that across the entire range of the species, that 10-day shift that we detected is really driven by individuals that were collected in warmer portions of Streptanthus tortuosis's range, or the jewel flowers range. And these divergent shifts could be driven or caused by a couple reasons. One, it's possible that individuals in warm regions are more sensitive to temperature than individuals in cool regions. Two, it's possible that individuals in warm regions have experienced a greater degree of warming. That would also explain this shift. Or three, it's possible that both of those things are happening at the same time. And so first, we looked at how sensitive individuals in both of these regions are to shifts in temperature. And what we found is when we look at the relationship between the day of year of collection and these temperature anomalies, which indicate whether or not that plant was experiencing a warmer or cooler than average year, what we see is that in both regions, individuals tend to flower earlier in years that are warmer than average. However, the response is much stronger for individuals in warm regions than cool regions. So individuals in warm regions shift their flowering date about 8 days relative to about 3 days for every 1 degree warmer than average year in cool regions. So this alone really could explain that divergent shift that we detected, but we also wanted to see whether or not the individuals in our data set were experiencing different degrees of warming as well. And when we look at the relationship between minimum spring temperature, which is the temperature variable in which Strophanthus tortuosus is actually flowering and fruiting, we see there has been warming in both regions. However, the degree of warming has been greater in warmer regions than in cooler regions. And in conclusion, what we found is that these divergent shifts in phenology really seem to be driven both by differences in sensitivity to temperature for individuals in warm regions, as well as differences in the magnitude of warming. So it appears that individuals in warm regions are more sensitive to temperature than individuals in cool regions, and they have experienced a greater degree of warming. And so this study demonstrates that we really need to consider that both differences in sensitivity to temperature and differences in the magnitude of warming could potentially affect phenology, and that responses to phenology might be more spatially complex than we previously thought, and it can actually vary intra-specifically. That is, it can vary within species. That means that predicting future changes in phenology might require having maybe region-specific phenological models in order to make accurate predictions. So with that, I wanted to thank some of my lab members that helped me through this work, as well as a lot of undergraduates who helped with the scoring of herbarium specimens, and of course, the collectors of Strophanthus tortuosus, for which none of this work would have been possible without them, and those are the top 10 collectors of Strophanthus tortuosus for the data that I used in my study specifically, one of which is Alice Eastwood. Thank you for listening. I think we do have a minute for a question. Okay. In the back, in the blue.
Okay. Started me? Okay, thanks. Yeah, I just wondered, do you have a good sense of how long any particular population of Strophanthus tortuosus flowers each year? Is it a long period? Is it a short period? Just wondering kind of like what the error is around that.
Yes, that is a great question. The flowering period can be relatively long. But one thing that I didn't talk about in this talk, because it's a shortened version of it, is we controlled for variation and phenological advancement among specimens. And the way that we did that was we counted individual buds, flowers, immature and mature fruits on every specimen, and we used that to come up with what we call the phenological index that ranged from 1 to 4. 1 being a specimen was all in bud, and 4 is a specimen all in fruit. And we basically used that value in our models to control for variation among phenological advancement in specimens. And so even though it has a fairly long flowering period, we were able to control for some of that variation when detecting these types of relationships. That's what most of the undergraduate work was, meticulously counting reproductive structures on our herbarium specimens. Yes, Dieter, in the back. So I believe the question is, has anybody tested the heritability of, and I think maybe you're asking heritability of phenological sensitivity? Yes. So people have tested it. It is heritable. Not a ton of work has been done with that type of work, though, but we would expect it to be heritable and to be able to evolve as well. Reed. Mm-hmm. Yes. Yeah, that's a great question. So the question was, at one point there were a lot of subspecies of Strophanthus tortuosus that were described in the Jepson manual that have since been merged back into just a single species. And we did look at that a little bit. We preserved some of the subspecies data that were on the sheets, and we didn't actually find any patterns in terms of phenological sensitivity and differences among subspecies. It was really driven by where those populations occurred and not necessarily, I mean, and there's some correlation between where those populations occur and the subspecies, but it was mostly driven by where those populations occur. So even populations in warm regions in Northern California and Southern California had similar phenological sensitivities. Do we have time for one more? Okay, go ahead.
Touching back on the flowering, thank you. Touching back on the flowering duration question, I was curious if there's any, if you think there might be a correlation between advancement over the years and flowering duration. Like, do you think it's possible that they're flowering for longer with greater advancement?
Yes, that is a good question, and I didn't look at that at all. And I think that it would be hard to assess flowering duration using herbarium specimens, and I can't really speak to whether or not there's a relationship between flowering duration and flowering time specifically. But people have beginning to look at, what's the effect of climate change on flowering duration, and there are studies that show that the duration of flowering has extended, and so it's possible that is also true for Streptanthus.
Thanks, Natalie. Our next speaker is Katie Pearson, the data manager for the Symbiota Support Hub, an organization that supports biodiversity occurrence databases. She has been the project manager for the California Phenology Collections Network, a herbarium digitization project from across the state led by Cal Poly, and where she is a curator. All right, we don't want extra slides. There we go.
Great. Thanks, Allison. And I'm so jazzed to go after Natalie because a lot of the stuff that I'm going to be referencing in the latter half of my presentation is using almost the same methods. And in fact, she was a co-author on it too. So luckily, I don't have to give all the background, but you'll be seeing some similar graphs, spoiler alert. So as Allison said, my name is Katie Pearson, and I'm in San Luis Obispo. I work closely with Jenn Yost, and today I'm excited to talk to you all about insights into plant phenology from new digital tools. So I'm going to be focusing on how there are new tools that can enable this type of research that Natalie has been talking about, and that we also did, so that more people can be studying these types of these things. So, as you likely know, our botanical knowledge is underpinned by herbarium specimens, and therefore by Herbaria. So I just want to give a big shout out to everyone who's worked in an herbarium and all the collectors, because this is really the basis of our understanding of where and when plants occur. And if you came to the session yesterday, you probably saw this same graphic a couple times. One of the reasons why these new tools that I'm going to be talking about have become available is because in 2018, the California Phenology Network was funded by the National Science Foundation. Jenn Yost was the lead PI of that, and I have been the project manager. And this is a collaboration of originally 22 herbaria. It grew to 28 herbaria to digitize herbarium specimens across the state. So the goal was to image, transcribe the label data, and georeference, that is, place a point on the map, for over 1 million specimens at the end of it. And then another piece of it was to score the phenological status of the herbarium specimens, and I'll talk about what that means in a moment. So as a result of the California Phenology Network, we created this data portal, the CCH2 data portal. It's easy to find, cch2.org. And it currently contains herbarium specimen data from over 4.5 million specimen records from over 70 collections. So these are collections from across the state and even a couple of partner herbaria that have a lot of California collections. So the CCH2 portal has some really slick user tools. It has specimen data and image searching, so you can go and look for all those specimens based off of the collector, the date, et cetera. There are mapping tools so that you can put those all on a map and visualize where those specimens are, depending on whether those specimens have been georeferenced. A little over half of the specimens in CCH2 have been georeferenced. If you want to help out with that, let me know. You can also create checklists, and I think we had a presentation on this yesterday, where you can create a checklist based off of a location or a species list that you've already compiled and you want to augment using the specimen data right there in the portal. So that can be really useful for a variety of things. And I don't need to go into too much detail about phenology, but if you just walked in, phenology is the study of the timing of life history events. So in plants, we often think about flowering or fruiting or sporing. I'm going to be talking about angiosperms here, so flowering and fruiting. And phenology is important for plants because phenological events can determine or affect fitness. So the timing of a phenological event can determine when that plant in that certain phenological status is going to interact with pollinators, with herbivores, with migratory birds that are dispersing the seeds, with climatic variables, so whether it's cold or it's hot or it's rainy, and that can all affect the plant's fitness down the road. So understanding those phenological events and how they may change with climate, with land use, with changes in distributions is really important for understanding the trajectory of those plants moving forward. And then very practically, phenology is important to understand and to know about as land managers, as researchers, as educators, as conservationists, because knowing when plants are flowering is important for timing your inventories, for one thing. If you want to go find a plant, you probably want to know when it's going to be out. If you want to collect it and identify it, you should probably know when the reproductive structures are going to be out. So it's just really practical to know when a plant is flowering and fruiting. It's also important to understand phenological events, or at least know when they're going to happen, because it can affect your restoration strategies. So if you have a widespread plant that has populations in certain areas, they may have different flowering times depending on when those plants, when the spring occurs in those regions. And sometimes plants can be locally adapted and flower at a certain time, and that can be genetically based. So if you're collecting seeds from one population that flowers 30 days earlier than another population, and then you try to mix them together and throw them in the field, you're still going to have two different populations that don't intermingle a lot. So it's really important to know when your source population is going to be flowering so that you make sure to sync those up. And then also very practically, if you want to go collect some seeds, you have to know when the seeds are going to be out. So having access to all this data is really important for all of us here. So in CCH2 and from herbarium specimens, we've been able to create some tools that help us collect this phenological data and make it available to everyone. And one of these ways is to collect phenological data from herbarium specimens from images. So there's this really neat tool now that you can bring up pictures of specimens and you can zoom in and you can look at the specimen and see whether there are reproductive features on that plant. And then you can use the scoring schema to say, yes, there are flowers, yes, there are fruits, or no. And so that's a way of scoring and collecting and storing those phenological data. But then also as part of this project, we developed a tool that scores phenology, or that you can use to score phenology from the label text. So herbarium specimen labels may have phenological data on the label, so written into the text. If it says flowers yellow, most of the time that means that there were flowers on the plant that the collector could see. And so we can use that database data to batch score lots and lots of specimens at one time because they all had flowers yellow in the description. So we were able to do this type of scoring schema for about 1.3 million specimens. So no, we didn't individually look at 1.3 million specimen images. We used some really neat tools. So as a result of all this scoring, these data are now available on CCH2, so you can search for phenological data, you can download phenological data, use it in research, make your own maps, whatever. Or you could use the phenological data visualization tools that we developed as part of CCH2. So if you go to the taxon page of any species in CCH2, provided that it is a flowering plant, there will be aggregated data for that specimen or for that species right there on the page. So you can see a nice little graph that tells you when approximately at least the specimens in our database have been collected as flowering or fruiting, and it gives you a good idea of when that happens. So I'm not going to go into a lot of detail about what you can do using herbarium specimen-based phenological data. There's been a lot of great research on this, and so if you want to read a review, scan this QR code. It's pretty comprehensive. Tells you about all the great things that you can do using herbarium specimens for phenological research. And today, I'm just going to talk about one specific example that we were able to do using the data directly harvested from the California specimens. And this was in collaboration with UCSB and all of these wonderful people. And you can read the paper in Madronio, and I'll have a QR code for that later. So in this particular research project, we were focusing on the California poppy, Eschscholzia californica, because it's iconic and very well collected. And we were interested in whether the flowering date of Eschscholzia californica is sensitive to interannual variation in temperature and precipitation. And then secondly, we wanted to know whether this sensitivity, if we found any, depended on long-term climate conditions. So kind of like what Natalie was saying, in warmer or cooler regions, is it more or less sensitive to temperature and precipitation? So I'm going to be showing you some similar graphs to what Natalie showed you, but if you weren't here, on the x-axis, you can see is where we're going to put our climate variable, our explanatory variable. And then on the y-axis is the flowering date. And so this is approximated by the collecting date of those herbarium specimens. And then the climate variable is created or derived from the date and the location data that is on those herbarium specimen labels. So we excluded anything that did not have location data, did not have date data, and we derived these climate variables from the PRISM climate group. So let's go into my first question. Are California poppies sensitive to temperature and precipitation? And I'm going to talk first about precipitation because the temperature is going to lead into question 2. So for precipitation, we found that the flowering date of California poppies is delayed with increased fall precipitation, but it's advanced with increasing spring precipitation. So it matters when those changes in climate happen in so much as it affects the California poppy. So that was interesting because you can't just say that rain causes earlier flowering or rain causes later flowering. It really depends on when that rain comes. It makes a lot of sense because if the rain comes earlier or later, then that's going to affect when that plant germinates, when that plant develops, and how long it develops. But it was interesting to see that using this data set. So before I talk about temperature, I want to go back to this second question of does the sensitivity to interannual variation depend on long-term climate conditions? And our answer to this, at least for temperature, is yes. In precipitation, it didn't matter whether there was more rainfall or it didn't matter whether there was more or less precipitation depending on how much precipitation is there normally or on average. But in temperature, flowering date advanced with warmer springs, but it depended on the long-term climate conditions at the site. So at cooler than average sites, we found that the sensitivity to temperature was increased, or it was more sensitive to temperature than at warmer than average regions. So in warmer than average regions, there was very little change in the flowering date of the California poppy, which is very interesting because it directly contrasts with Natalie's discoveries with Streptanthus tortuosus, meaning that there's definitely some interspecific differences, and as you can see from this graph, intraspecific differences too. So on the x-axis is the more or less than, or warmer or cooler than average temperatures on the x-axis, and then on the y-axis is the flowering date. And the blue line shows you the sensitivity to temperature in cooler than average regions, and then the red line is warmer than average regions. And you can see that the warmer than average is pretty flat. You're not seeing a huge sensitivity to temperature in these warm regions. So what could this mean? Well, there's a similar conclusion to what Natalie was saying earlier, in that if you're getting an estimate of the phenological sensitivity of a species, then you probably need to understand that phenological sensitivity across the whole range if you're trying to make predictions for the species as a whole. So single phenological sensitivity values derived from a single location may not accurately predict sensitivities across the range. So if you're doing a small-scale phenological study, you have to understand that you might not be able to apply those same results to the population in a warmer or cooler region. And then secondly, and this is specific to the California poppy, but we're wondering, and this is a hypothesis, whether the lower sensitivity in warmer climates could reflect developmental limits or evolutionary constraints of those populations in warmer than average regions. It could be that the California poppy just cannot flower any earlier in these warmer regions. And that might mean that its length of time living at those warmer regions may be coming to an end. So it could be that the California poppy needs to shift into cooler areas if it's going to continue to track climate change. So this is just one example using one species and about 900 specimens that used phenological data from CCH2 to apply it to real-world understanding of climate change effects on this species, the phenology of this species. But we have, as I said earlier, 4.5 million specimens, and at least 1.4 million of them have phenological data associated with them. So as you can imagine, the opportunities to understand phenology are very, very, very great. So I encourage everyone to go to cch2.org and hunt around, explore your favorite taxa. And if you're interested in doing phenological research, you have this giant data set right here. So thanks to all my collaborators and really all of the herbaria on this project for making this possible. And if you want to read the paper, there's the QR code to go look at that. So thanks to everyone, and I think I have time for questions. Any questions? Yes?
Can you pull data for a group of plants, like put in an area and then get a group of plants when they would, when you could collect seed and when they would flower?
What you could do is you could do a map search based on like a point radius or whatever, and then you can create a checklist from that, and you could look at the phenological timings of all the taxa on that checklist. It won't be able to say like all of the plants in this area generally flower at this time, but you could look at them per taxon.
So I can just put it in by put it in by a map area.
Yeah.
Okay, thank
you. If you go into search specimens map search, then there are tools there.
Thank you very much.
Yeah. Other questions?
For both you and Natalie, there's a special elevation range around the Central Valley where snow used to lie on the ground most of the winter, and now it doesn't. And that line has moved up from about 3,000 feet to 4,700 feet. So there's like a big climate effect at that elevation. And you guys both, you and Natalie both have enough specimens from elevation ranges that might be affecting some of the results, and you might have enough points to actually look at that.
Yeah, that would be really interesting to look
I
at.
think the climatologists call it the banana belt.
The banana. I think there was one more question. Yeah. back there if we have time.
I'm interested in putting together local plant lists, and I've been working mostly with experts in their areas. Do you think that your coverage now is complete enough that if you just went into a new area that you would get a high percentage of all the species that are in that area?
Oh, yeah. Yeah. I think that you would be able to create a probably not comprehensive checklist, but pretty close.
Especially for the common ones that people would notice.
Mm-hmm.
Right.
Yeah, it can depend on how many specimens are geo-referenced, so you might have to do a couple of searches and combine them all together.
Okay.
But you can usually create a pretty close to comprehensive list.
Thank you.
Great, thanks. And if you have any questions about CCH2, if you go on and you're like, I didn't understand this thing, I'm the person to talk to. Actually, there are a lot of people who know a lot about CCH2 here, but feel free to ask me afterwards.
Our next speaker is Lacey Benson. She's a graduate student at the Lambrecht Plant Ecology Lab. Let's see, let's make sure there we go. At San Jose State University, and assistant curator of pteridophytes at the Carl Smith Herbarium. And she's currently investigating western sword fern leaf morphology and fog interception in the coast redwood forest, which is what we're going to hear about today in part. And Lacey is a Bay Area native. She has a passion for preserving and restoring native landscapes to enhance biodiversity and cure plant blindness. So at the Smith Herbarium at San Jose State University has 26,000 specimens, 18,000 vascular, 8,000 bryophytes, and 16,000 are digitized and 14,500 are imaged. Thank you, Lacey.
Good morning, everyone. Can all my fern fanatics hear me in the back? Awesome. Okay. Thank you for that introduction. My name is Lacey Benson, and it's great to be here to share with you some of the things that I've learned studying western sword fern populations across the coast redwood forest ecological gradient. I don't have results to share just yet, and I'm in the process of collecting and analyzing a lot of my data through this winter. So in this presentation, I will go over the concept, my study design, preliminary data, and then things that I've learned along the way. So I'm happy to present my project, a morphometric analysis of western sword fern, Polystichum munitum, pinnae and pinnae scales across the coast redwood forest ecological gradient. But before we get started, I'd like to acknowledge and pay tribute to the land on which this project took place. So take a moment to recognize that you are on native land and increase your awareness of the rich and diverse peoples that inhabited and still inhabit this land. So before you is a list of indigenous territories on redwood country. The coast redwood forest, if you're not familiar with it, spans a 40-mile wide and 450-mile long coastal strip from southern Oregon down into Monterey County. Along this strip, rainfall decreases from north to south and more abruptly from the coast to inland. Fog mostly occurs in the months of June through September. And researchers at UC Berkeley found that 13 to 45% of annual water input in the redwood forest ecosystem is supplied by fog. Summer fog helps alleviate drought stress in plants by increasing relative humidity and lowering rates of evaporation and transpiration. Summer fog is an important input when rainfall is scarce, specifically in the summer. Plants in the redwood forest have adapted to intercept and absorb atmospheric moisture through the leaves, which is called foliar water uptake. Unfortunately, due to increasing inland air temperatures and rising ocean surface temperatures, maritime climate, or fog, has decreased by 3.5 hours per day over the last century. However, more information on how this variance is occurring needs to be done. Okay, So by studying morphological as well as physiological changes to ferns, scientists can get a more rapid understanding of how community dynamics and slower growing species, such as the coast redwood, will be affected by future changes. Ferns display signs of shifting climate patterns through their leaf traits, such as number of fronds, size of their fronds, and foliar water uptake capacity. Polystichum munitum, or POMO as it may be referred to in future slides and by me, depends on foliar water uptake during the rainless summer months to maintain an optimal water status in its leaves. Foliar water uptake and leaf water retention varies geographically. Lim and Dawson researchers found that the southern populations of POMO, like those at Big Basin State Park, had no foliar water uptake capacity. But they did have high leaf water retention ability. So these variations in cuticular permeability in the literature have been attributed to environmental variability across the gradient, as well as leaf cuticular properties and leaf surface roughness. But it is assumed that foliar water uptake is facilitated via diffusion across the cuticle. However, more research into this mechanism of this pathway will improve our understanding of the observed variations in Polystichum munitum populations. Additionally, research has found that leaf area varies geographically. So Free researchers have found that frond size and abundance increases along the northern latitudinal and precipitation gradient. So for my study, I have 3 objectives. First, I want to compare pinnae size traits, that's length, width, and the length-width ratio, to precipitation and fog frequency data. And secondly, I'm going to quantify pinnae scale density and compare this with HOBO data loggers and historical climate data. And then lastly, I want to collect, which I have done, describe, mount, and digitize POMO specimens as well as redwood associate species that I have collected from 2019 through 2021. And these will be placed in the Carl W. Shar Smith Herbarium at San Jose State. So in December 2019, I placed 5 external HOBO data loggers at 5 different locations across the coast redwood forest, which you can see in the map on the right, highlighted by the yellow boxes. These were placed at sampling locations, and these data loggers collected fine resolution climate data for every hour for 12 months, and they were collecting temperature, relative humidity, and dew point. As of December 2020, these loggers have been collected. However, the data logger, which was at Big Basin, I could not recover the data because it was burned in the 2020 CZU fire. And then additionally on the map, you can see the 24 different locations where I sampled POMO vouchers from, which are highlighted by the yellow circles. Okay, so since 2012, Fern Watch has monitored POMO's response to climate at 11 different locations. So I've used their map, but these are not all their locations. These are my locations. This provides long-term climate data, or long-term data which shows the forests which are most impacted by drought. Because POMO is an important indicator of drought stress in slower growing species. So the location data for both of these bar charts is distributed from north to south, where the wetter northern range receives around 68 inches of rainfall, and the drier southern locations receive around 21 inches of rainfall. The one on the top left, you can see as the drought was in full effect between years 2012 and 2013, which are the bars in blue and green, POMO was able to avoid drought by reducing its leaf area by approximately 1/3. This meant that ferns were creating fewer new fronds and had fronds that were smaller in size. Just comparing both of these charts with my preliminary data, you can see overall how climate can be a limiting factor in vegetative growth. So fronds and leaf sections in northern climates are longer and more numerous, while those in the southern distribution we find are shorter and have fewer fronds per individual crown. So for my data collection, aims 1 and 2, I'm using digitized ascensions in image J, and then my pinnae will be measured at mid-rachis. In the image on the lower left, you can see a pinnae. Also, there's a two-dimensional image for your reference on the top. The pinnae length was measured in yellow, you can see, along the costa to account for curvature of unique pinnae. Also, the pinnae width in red was measured immediately past the auricle or the thumb on the pinnae at a right angle to the costa. So these measurements that I'm taking will be compared using principal component analysis, and I'm going to compare them against the field collected regional weather data and historical climate data. My second measurement, scale density, will be calculated using leaf area and a scale count of 5% of the leaf surface in image J. So I've created a nice little diagram on the right side. You can see both surfaces of a pinnae and different types of scales you might find on a sword fern pinnae. On the upper surface, I found most of the scales along the costa, and most of those scales were very long, and then they had a kind of spidery, feathery formation where they attached. And then on the underside, we had a more flat, oppressed type of scale along the costa, and then on the leaf surface, there's these beautiful kind of wide, feathery scales. And you can infer all kinds of things about how they might intercept or aid in moisture retention on the leaf surface from these. Oh, sorry. I can go back later. So my aim 3, collections and ascensions. My samples included in this analysis came from a variety of sources. In 2019, we collected 19 pomo over the summer and fall. And then over the summer and fall of 2020 and 2021, nearly 70 pomo were collected from less represented regions. During the pandemic, I quickly learned how few pomo specimens were digitized that would fit within the parameters of my study. Those that did, I included were 25 sheets obtained from CCH2 and other partnered herbarium sources. And then lastly, I included almost 20 sheets from the California Academy of Sciences, which were through digitization requests. My samples that I collected were from old growth and second growth redwood forests along alluvial terraces within 50 meters of a watercourse. And then my field collections were focused on identifying type specimens, hybrids, ferns of different developmental stages, as well as fern and fern allies. And then to date, over 130 collections have been made, which include 13 different fern and fern allies, as well as 16 understory redwood associate species. So, since I don't have data yet, I can share with you my expected results. For character measurements, we predict that southern populations are expected to have smaller pinnae, while populations collected from the northern latitudes will have larger pinnae. This is a positive relationship with pinnae size and precipitation. And then for scale quantification, southern populations are expected to have a lower density of pinnae scales, while those from the north will have much greater density of pinnae scales. And so we predict that pinnae scale density will be inversely correlated with precipitation. And then finally, for our pomo collections for vouchers, we expect that these collections will be useful for future studies in investigating the plasticity or genetic differences between Polystichum munitum populations within the redwood forest. So some lessons from the field and the herbarium. So when you're collecting Polystichum species, be aware of hybrid zones. These are habitats where similar taxa can overlap, such as ecotones, like rocky outcrops meeting loamy music sites. These are, ecotones provide sites where hybrid populations can occur. If you look on the bottom, you can see an example of two different indusia coverings of Polystichum munitum and then Polystichum imbricans, subspecies curtum. They can look morphologically similar, and then they can also inhabit the similar kind of habitats. So keep that in mind when you're collecting sword fern. Also, you should collect the entire frond. So the image on the left side, you can see this frond is taller than I am. Good luck fitting that on one voucher. So if you can't collect the entire frond, make sure that you're measuring it and you're including these measurements of the entire frond, the stipe, the rachis in your species description. Also, you should look at the basal scales. So in the upper right, I have some basal scales there. Those are important for species determination between the Polystichum species. So keep that in mind when you're also collecting sword ferns. And then you should bump up your descriptions. You should include habitat features, dominant tree species, visually estimate canopy cover, herbaceous understory composition, and then try and collect fertile fronds in order to check the indusia margins, and then also you can use the spores to estimate genome size or see if you have a sterile or hybrid fern. So these next two slides, I felt like they're important to include because it's where my field observations and my research has taken me. And these are still things that need to be addressed. So if anyone's interested in doing genetic work or setting up a monitoring program, these would make great projects. Western sword fern is adapted to survive drought. However, researchers have found prolonged water deficits coupled with insect damage can hamper frond survival. Researchers have documented cases of community-level dieback in southern populations during the drought in 2014, resulting in reduced population density and higher susceptibility overall. So in the top two images, you can see damage caused by thrips, which are sap-sucking insects. One is from Andrew Malero from November 2020. Another example of community-level dieback I saw out in the field when I was collecting firsthand in Big Sur. I saw an entire hillside of sword ferns with crispy leaves and significant frond damage. I would like to revisit this location in the spring and monitor for new frond growth. Secondly, there needs to be mapping of genomic variation across the range of the POMO complex. Earlier work has demonstrated that POMO is predominantly outcrossing, exhibiting greater genetic diversity within populations than among. And one example I tried to include in my vouchers was the different Polystichum munitum individuals growing next to each other. In the top right image, you can see the morphological variation in indusia color. These were two different individuals growing right next to each other. And the one on the left you can see is kind of a darker coloration, which I dubbed the chocolate chip indusia. So understanding the genomic variation with the within the POMO complex will help us understand the degree and directionality of gene flow among related taxa. Secondly, I tried to include in my vouchers different taxa. So Schmempecker et al. Identified that the diploid Polystichum munitum and the rare endemic Polystichum dudleyi are progenitors of the rare allotetraploid Polystichum californicum. And you can see from left to right, there is P. dudleyi, and then the allotetraploid californicum, and then on the right, Polystichum munitum. So additionally, identifying the pathogen, genetic, and environmental factors that are contributing to sword fern die-off in Washington state could help us prevent and control its spread into California. So future questions from my research are, why do pomos in the southern range have a limited capacity for foliar uptake? Do scales aid in foliar water uptake, and is this a plastic trait? Additionally, how will pomo populations in California be impacted by sword fern die-off phenomenon, which has been observed in Washington? And then lastly, will pomos in the southern extent of this range persist, adapt, or be replaced altogether by drought-avoiding species of ferns? And I want to thank my advisor, my project funding, my field assistants, and my mentors. So with that, I'll leave my 1 to 2 minutes for questions. And feel free to contact me. No questions. Oh, yeah.
Does the water uptake occur just on the living leaves or also on the dead leaves? They tend to make this big skirt of dead leaves around them.
It's just on the living leaves, yeah. So that variation was across the entire ecological gradient, and they found the ones in the south had no foliar water uptake capacity, but the ones in the central distribution had the highest ability for foliar water uptake. So this variation is still needs to be understood. Thanks, everyone.
Thank you, Lacey. Our last speaker of the session is Lua Lopez, and we'll cue her up. Lua is the curator at the Cal State University San Bernardino Herbarium and an assistant professor there at, is it CSUB? How do you?
CSUSB.
CSUSB, okay, in the biology department. Her research focuses on how species respond to their environment and the changes occurring over time. To do so, she uses common garden experiments with modern populations with genetic analysis from herbarium specimens. The CSUSB Herbarium was founded in 1969, 4 years after the campus opened. The collection mostly includes specimens from San Bernardino and Riverside counties, approximately 4,000 specimens, all digitized and imaged. All right, here's Lua.
Hi, everyone. Can you hear me correctly? Awesome, thank you. Okay, so today I'm gonna be talking about what kind of things we can do with herbarium specimens in terms of like adaptation studies to environmental changes. The talk is gonna have two parts. One where I'm gonna focus on some new wet lab techniques that I've been developing with help of some collaborators, and the second part more about the genetics part of it. This is the California Native Plant Society, and I'm going to be talking about Arabidopsis thaliana, which I know it is introduced in California, but bear with me. I think I can tell you some interesting things that can be applied to any other species. So I don't have to convince anyone in this room that the environment that we are living in is drastically changing. I just put in there a graph for temperatures. We've been seeing these kind of graphs during these past 2 days, but heat is not the only thing that is changing in our environment. We are also talking about drought or flooding. So the question is, how do organisms, and namely plants, can respond to these environmental changes? And in theory, when you try to investigate how things have changed over time, we focus normally on current populations, right? The problem is that, can you see? Oh, you cannot see them. Trackpad. Okay, whatever. When we focus on now, current populations, we're only obtaining a very small snapshot of everything that has happened in the evolutionary history of that population or even in a species. And the problem is that different genetic signatures, the same genetic signature can lead to different evolutionary processes, or it can be consequence of different evolutionary processes. So we don't know what has happened there on the interrogation mark pattern. If we are talking about a species that have conservation interest or even invasive species that we are trying to control, it is really important that we make accurate conclusions. One way to solve this issue is to do long-term experiments, but we all know that these are really expensive, not only in terms of money, but also in terms of personnel. It can take the life of several researchers to get meaningful results. So what can we do in this situation? Well, ta-da, we have an awesome opportunity to use Herbaria specimens to have long temporal and spatial trends. And not only that, I don't only get the spatial and the temporal pattern, but herbaria have like a massive amount of specimens that I can relate on. I think the number for like the global herbaria effort in terms of specimens that are stored around the world is like about 350 million. And we are talking not only about things that are still alive, we're talking even about species that have been extinct, so we can even understand what has happened that led to the extinction of those species. So basically, if we put all this together, we got ourselves just a time machine, which is really cool because I can directly observe what kind of genetic changes happen over time and over space in association with environmental changes. But the thing is that studies using these kind of samples are still not that common. We're getting there, but most studies have relied on PCR-based techniques, like just focusing on small DNA fragments. And the reason for that is because DNA that comes from museum specimens, it's highly degraded and damaged. So over time, the DNA is gonna get fragmented, and on the edges of those fragments, there's gonna be an accumulation of C to T inversions. So how do we work with this kind of damaged DNA? It also comes in small quantities, by the way. The thing is that over, I don't know, the past 3-4 decades, studies on humans and mammals are very well established. New wet lab techniques, new bioinformatic tools have also allowed that this type of studies in other taxa will keep increasing, like in fungi and in bacteria. And that graph stops in 2014, and I have to say that things have gotten better since then for plants, but what you can see there is that plants are nowhere to be found. Like the line, it's very thin, it's not very obvious, but there's this thin green line. Why? What's going on in here? What happens with plants that is making things so difficult? Well, the problem is that plant tissue is soft. It's not like bone structures or like shells where DNA can be preserved longer. So some studies have pointed out that DNA stored in tissue for plants will degrade even faster than the one that is stored in bones. On top of that, if you have ever had in your hands an herbarium voucher, and I'm pretty sure everyone here has at some point, you can see that bacteria and fungi will feast on the tissue, especially if the preservation conditions hasn't been good. So you end up extracting a lot of DNA that is not from the plant that you really want to use. But not all people working with herbarium samples think that the DNA is so degraded. In fact, some people have pointed towards this idea that maybe the DNA on herbarium specimens is still it has a pretty good integrity. So what I tried to do with this, and I've been working on this for the past 5 years, so I feel like this is right now kind of like my child project, is to focus on understanding how this DNA behaves and also trying to study what has happened to Arabidopsis thaliana over 200 years in terms of like adaptation to climate. And why Arabidopsis thaliana? Well, because it's a model species. We have so much information on it. And when you want to test any kind of new method or you want to optimize on all one, a model species is the best way to go. On top of that, Arabidopsis thaliana, it's an annual species that is native to Eurasia and North and East Africa. So that covers an immense amount of different environments, which makes it ideal to study how plants will respond to environmental changes. Finally, it's diploid, mostly selfing, and it has a beautifully sequenced and annotated genome that makes my life way easier. So as I said, the talk will have 2 parts. The first one is to tell you a little bit about how we did some testing and optimized some wet lab protocols for whole genome sequencing in herbarium specimens. And the second part, I will talk a little bit more about the genetic responses and the population structure of these species. So let's talk about wet lab techniques. I told you that ancient DNA, it's kind of tough to work with, low amount, highly degraded. So I did a pilot study, and I focused on 11 samples that each of them is represented in a bar. And I used a previously published SNP matrix for this species, just to kind of have an idea of how much of it I was covering in my samples. And here you have the ratio or the amount of missing SNPs. So basically, things that I was not finding in my samples. This is not acceptable. Like, if I want to do any kind of genetic work, this amount of missing SNPs is not going to take me anywhere. And also, there was like not a pattern in terms of like, okay, I always have like this amount of missingness. No, each sample behaved differently. So I had to go back and think about this. And I realized that a lot of my samples had many, many reads that they did not belong to the plant DNA. They belong to bacteria and they belong to fungi. So that is pointing towards this concept that I have a lot of exogenous DNA. And what I want is my plant DNA. And I told you that DNA is highly fragmented in herbarium specimens. Well, yes and no. I did a very old school agarose gel. I love agarose gels. I think they're very informative. Just to look at the pattern of fragmentation of my samples. And what you can see here is the 500 BP sample, ladder band, sorry. And yes, I do have a lot of fragmented DNA. Like, a lot of it is down here. But what about all this stuff up here? I also have a lot of DNA that is not fragmented. And here's the trick. When we work with ancient DNA, we do not fragment DNA because we assume it's so fragmented that we go for library prep and sequencing all straight without doing that fragmentation step that we will do with modern samples. So after seeing this, I decided to create kind of like a hybrid protocol. I added a really, really short shearing step that was targeting the genome size of this species. And then I did the library prep and the sequencing again and the SNP copying. And this is what happened. In orange, you can see now my SNP calling. The amount of missing SNPs has significantly decreased for most samples. And not only that, it has stabilized. This is something that I can work with. So the question here is like, okay, why adding that shearing step has helped so much? Well, I teamed up with someone who's here in the audience. Shout out to Patricia, thank you. And we selected around like 100 samples that I did both with that hybrid protocol and the ancient traditional protocol. And our first question was, can I still detect signs of ancient DNA? Because when you want to publish this, they will ask you for proof that this is indeed ancient DNA. And that is how we look at ancient DNA. Like, we need this increase of C to T inversions at the edges of the fragments. You can see that the samples that were treated as ancient DNA in black, they do show higher damage, but the shear ones do still show that damage. So we are good. We can still go ahead with this, and people will believe us, this is ancient DNA. So what the heck is happening here? What is improving my results? We have two hypotheses. It could be that we are covering a larger part of the genome because we are targeting fragments that previously we weren't capturing in our sequencing, or it could be because now we are going to have a higher sequencing depth because we are increasing the plant DNA out of our samples. When you look at the merged reads here, that means when I have forward and reverse and I put them together for my analysis, the fraction of the genome that I'm covering is the same, either I fragment it or not. But there's always a certain amount of reads that you don't get both forward and reverse, but you don't want to throw this away. Again, ancient DNA comes in really small quantities. And this increases the coverage of my genome around like 10-15%. So this kind of suggests that we might be covering more of the genome, increasing our SNP calling. We are now working on figuring out if the sequencing depth is also something that is playing a role, because it doesn't have to be A or B, it could be A and B together. But more results to come. We are working on our manuscript and we are hoping to put a preprint out, hopefully January, February. So just stay tuned. For the genetics part, that second part of the talk, I have over 500 samples right now collected across its native range. And you can see here on the map where all the samples come from. They have been collected during the past 200 years, which is like really good in terms of like when Industrial Revolution started and how it kept going, and now like this big moment of climate change that we are all experiencing. So in terms of like global patterns of genetic diversity, well, the first thing that I did was to look at how the samples cluster. And this here is an admixture graph. Every bar is one individual, and the colors will indicate genetic groups. The samples, all of them, have been put in here. So the first thing to say is that if I was seeing a big difference in terms of time, samples won't cluster geographically. They will cluster temporally. But they don't. They cluster geographically. So this green belongs to Africa, so these samples here, and also Morocco and some South Iberian ones, and then the rest of Europe and Asia. If I keep increasing the number of clusters, then the Asian samples will diverge from the rest. And this is what we have seen previously in other studies. But this study covers some new geographical regions that hadn't been previously sampled, which is here, the sub-Saharan Africa. We had samples from up here, and we knew that those were like a very different genetic group, but now this adds a whole new different group to that. So it looks like we have some relic evolutionary lineages in East Africa and the Southern Iberia and North Africa. And the rest of the population structure is the same one that had been previously reported for the species. And as I said, these African samples are new to the global diversity pattern. And what we can see is that for these samples, we don't find that geographical clustering. What we find, it's a clustering based on the massif, the mountain where they were located. So there's like very little gene flow between all these big mountains. We're talking like 4,000 meters high, like Bale Mountain, Simeon Mountains. So there's a big genetic isolation between them. There is some samples that do cluster together, so that will be Tanzania, Mount Zuza, and some South African samples, but the pattern focus on those mountain massifs. And then I can look at those temporal changes in smaller regions. So that map that I show you, some of the regions I have a very deep sampling. One of them is Norway. So what I can do is to use time as if it was my phenotype and look at what kind of genetic changes I've had over time. And this is because it's Arabidopsis thaliana and I have that beautifully annotated genome. So in Norway, I have detected two peaks, one in chromosome 1 and one in chromosome 5. And for example, the peak in chromosome 1 is associated with some genes related with response to biotic stimulus and defense response to. On chromosome 5, the elongata 3 gene pop up, which is associated with an arrow leaf phenotype and reduced root growth. So these are still preliminary results. I'm working on this. We are trying to obtain a general picture also for the whole geographic range, and then a more in detail picture for the regions that I have closely sampled. So overall, at this point, if someone would ask me, I would recommend to check every single of your samples for their DNA pattern. If you have DNA above 500 VP, go ahead and do that small sharing step. This applies to any species, not only Arabidopsis thaliana. I forgot to mention it, but this study brings in a new whole SNP matrix that covers genetic diversity that wasn't previously covered, that African samples. We have 9 million SNPs. So anyone who wants to use this can use it to screen for SNPs on Arabidopsis samples. We found that population structure was stable over time. Things didn't cluster based on moment of collection, but rather based on geography. There are two main genetic lineages, that African, South Iberian one, and then Asia and Europe. There are local genetic temporal changes. I still have to do more work on this, and there is a lot more to come. This project is massive. It has taken a whole new level, so I'm hoping to complete it pretty soon, but we'll see how that goes. And of course, I couldn't have done this without all the herbaria that send me samples, that allow me to go there and take my own samples. Some really amazing people that helped me with this. I started this project as a postdoc at Lasky's lab at Penn State, but Patricia, who's here in the audience. And thank you all of you for listening to me, and I'll be happy to answer any questions. Thanks.
Hi, thanks. My question is, like, what do you think the cutoff is for an herbarium specimen to need ancient DNA techniques? Because we do a lot of normal sequencing on herbarium specimens as well.
It depends on what you're going to do with it. I think that if you're looking to do more like PCR-based, just like focusing, that was making noise, a few genes, you might not need to do that shearing step because obviously you will have your primers. But if you're looking to do whole genome sequencing or rat sequencing, like something that is going to capture more, I don't think we have a cutoff at that point. I think definitely you need to assess the fragmentation pattern of your sample because, let me go real quick to this. Here. I don't know if you see the time of collection, it's in there. There is a sample from 2010 that has like a 48% mapping, and one from 1982 that has a 78%, and then this one from 1990, a 13%. So I think there's something that plays a massive role in here, and it's how samples have been stored over time. Like not every single place has the same resources, so definitely if samples weren't in the right condition, then bacteria and fungi are going to be much more common and they will degrade faster. So I don't have a cutoff. I will tell you, do every single one of them, just run them and check fragmentation patterns.
Yeah, 100%. Just, were you actually having a completely clean lab?
Yes, I did this in a dedicated ancient lab, but I don't have that right now, and I've been doing DNA extractions in my regular lab, and it works. The only thing is that you need to have kind of like an isolated space, so what I've done is to buy like a very good PCR hood that I can sterilize and clean, and it's completely covered, the one that you go just with your hands in there, kind of like chemical cabinet. So that way that space is locked. I currently work in a place that we don't have so much funding, so I had to become a little bit more creative with how to do this.
Yeah, absolutely, yeah, and I think it's really great to make these techniques more accessible to non-companies.
Yeah.
I have to ask one quick question. Are you working on California Arabidopsis?
Okay.
I don't know about others, but I definitely see them in weedy situations. I'm assuming they're from Eurasia, but sometimes I run into little populations in the mountains, and I'm kind of wondering about your Africa results.
I just got NSF funding, and it's specifically to work with that. I'm going to be looking at genetic mechanisms that allow species to migrate to higher altitudes as a way of escaping climate change. And the model organism is Arabidopsis, because if you plot herbarium records here in California for Arabidopsis over time, you see that increment in altitude. It could be a bias of how things have been collected, but because now I have this SNP matrix with all these good records from the native range, I can literally look at if I have new mutations that facilitated that increase, or if it's a standard genetic variation from some of these altitude regions. So yeah, I just got NSF funding to do that for the next 3 years. And I'm going to be hiring a postdoc. So if someone is around here, there will be a 2-year postdoc to work with me on this. Thanks.
Thank you, Lua. So that concludes this session. And there's Mike Simpson and Jason Alexander are going to be talking in the fur room tomorrow afternoon about more on this subject in case you haven't got enough. Thanks, everybody.